Here I sit down with origin of life researcher Ram Krishnamurthy for a conversation that we thought would be mostly about two of his new papers, but ended up becoming a broad overview of origin of life research and a deep dive into the nature of the scientific process itself. How can science, a discipline obsessed with direct observation and careful skepticism of any unfounded claims, how could such a discipline even try to study the origin of life? A process that was likely very messy and happened billions of years in the past. This was not a normal scientist interview for me. Ram and I have a long history. We met almost 15 years ago when working for the Center for Chemical Evolution. My role at the center was to teach the public what the scientists were up to. Ram recognized the importance of this, took me under his wing, and he ended up becoming the most important mentor in my career. Origin of life research is an extremely diverse field. In it you find geologists, chemists, biologists, physicists, mathematicians, engineers, planetary scientists, computer scientists, astrobiologists, and more. All of them are involved in this work. It can be very chaotic, very difficult to sort through. On the molecular side, you'll find some people working with really messy chemistries, the stuff in meteorites, hydrothermal vents, and volcanic pools. Then you have teams of people like Ram looking at specific chemical reactions, usually far cleaner chemistries, in a pristine laboratory setting. Much of our conversation goes over the strengths and the serious limitations of this cleaner type of research. What can and cannot be honestly understood about the origin of life when looking at reactions in a pristine, round-bottom flask? We talk about Ram the naysayer. Ram does have a bit of a reputation in the field. You may have heard that the role of a good scientist is to poke holes in their own work and the work of their colleagues. The role of a scientist is to falsify hypotheses. Obviously, that's not the only role of a scientist, but it's one that Ram has fully embraced. One of his recent papers is exactly this. He discovered a pretty big flaw at the heart of our thinking on the origin of ribose, the sugar found in RNA. Discoveries like this are frustrating. It might end up meaning we need to find an entirely different pathway for sugar production at the origin of life, but this is how good science works. We then talk about Ram the pathfinder. The building blocks of life, amino acids, fatty acids, nucleobases, and sugars, these building blocks themselves are complex. Most environmental chemical reactions that produce these things abiotically, they tend to produce them in low concentrations and with a lot of other junk. This issue is commonly known as the tar paradox. A common theme in Ram's work has been to discover and catalog natural sorting mechanisms, natural processes that pull these molecules from their noisy backgrounds, allowing them to, quote, find and react with each other. One of his new papers documents just such a process. And of course, in this conversation, we do stop to briefly talk about the elephant in the room. What happens when science and religion collide? Without further ado, here is my conversation with Professor Ram Krishnamurthy. This morning, I was curious. I don't know if you mess around much with AI, but I was curious how it would describe you. Because, we've known each other now for
Yeah.
something like 15 years.
Yep.
Or
Yeah, 15 years.
Yeah. I would introduce you as an origin of life chemist. It describes you first. It didn't even mention that actually. In its first thing, it said synthetic organic chemist. I'm curious. Is that how you would describe yourself too?
Yes, because that's what I do, and all the problems that I try to tackle using synthetic organic chemistry is inspired by origins of life questions.
Right.
So when people rightfully criticize my work saying that, oh, you use, pure chemicals, or you use, unrealistic conditions that are not prebiotic, I said that's true, because my work is prebiotically inspired, not prebiotically plausible.
Yeah.
Again.
Yeah, so we're going to dive into this. I mean, I think a lot of people don't understand that maybe even most of what we do in science is we debunk our own ideas, or we disprove hypotheses. What the reason I brought up the synthetic organic chemist thing is, I'm a science communicator, and it's so funny to me that the way that terms are understood inside and outside science, and
Yeah.
this phrase, synthetic organic chemist, that just does not compute to the public.
Yeah, because you don't put synthetic with organic when you go to the produce section and say, oh, this is an organic apple. That's a synthetic apple. How can be
So
synthetic be organic?
I think most people think of synthetic as like fake leather, right? Plastic
Yeah.
leather.
Mhm.
Anything synthetic is what you worry about maybe being in your tap water, right?
Yeah.
And then organic is thought of as healthy or pure.
Mhm.
So
Yeah.
just describe real quickly those words in actual chemistry.
So let us start from the word organic. When you have organic chemistry, what it means is you are dealing with compounds that contain carbon, hydrogen, nitrogen, oxygen, phosphorus, and possibly sulfur, what we call CHNOPs. So any compounds made out of those elements are considered organic. And so any person who works with those chemicals made out of these CHNOPs, that person is called an organic chemist.
Mhm.
He works from the chemistry portion of it, trying to work with these atoms or the combination of the atoms molecules to put them together in a fashion that was not intended or seen in nature. And there comes the synthetic portion. So when you are a synthetic organic chemist, you are working with organic molecules and organic building blocks and creating chemistry in the lab, which becomes synthetic. So you are a synthetic chemist. People say, oh, I'm a synthetic chemist. They leave out the organic sometimes. And a synthetic chemist generally is associated with synthetic organic chemistry. But you can also be a synthetic chemist and do inorganic chemistry, where you can do a lot of materials chemistry. So you can work with, metals, metal oxides. Then there is the combination of metal organic frameworks called MOFs. So
Yeah.
there are a lot of flavors in synthetic chemistry, ranging from pure synthetic organic chemistry to pure inorganic chemistry and anywhere in between. So
Yeah, so when another chemist hears that you are a synthetic chemist, they know that you're able to synthesize new molecules in your lab. That's one of your
correct. Yeah,
expert. And when they hear that you're an organic chemist, that means that your expertise is in the molecules that are commonly found in life,
that's right.
carbon-based molecules.
Yeah, and
Are
because the
are your jam.
organic chemist is divided into at least two major branches. One is what we call total synthesis, where you focus on synthesis of molecules, which is what people would use for, pharmaceuticals, drugs, etc., etc. And then the other is methodology, where you develop methods in order to achieve the type of synthesis that you want to do. For example, there'll be a reduction method that you want to do to make a compound more effective, or an oxidation method, and you try to find out what sort of methods would work best for this molecule, because each method sometimes, it'll work great on one molecule but not on another for a different reason. And then you have to find out, oh, how do I change conditions? So that is a methodology development, and the other is the synthesis development. So when you say you're a synthetic chemist, people would understand that you're making small molecules for the purpose of, bettering the drug candidates, or making molecules that would help alleviate certain sufferings, those kind of things.
Right. Right. You were talking about earlier about how there's a lot of people with this question.
Yeah.
How is it that by studying synthetic molecules in the lab, pure, often highly pure or close to pure
Yeah.
concentrations of stuff, how can that tell us anything about the origin of life? Because as an origin of life researcher, really what you're doing is you're being a historian. You're trying to figure out what happened, where did it happen, how did it happen, and in this case, billions of years ago. How can anything we do in the lab reveal anything about that in any way? How is
Yeah,
it even relevant?
it's a very valid question, and it needs to be asked every time, not just once. It's going to be a perennial question, and therefore this perennial question needs to be kept in mind, especially when we make claims. Okay,
Mhm.
that's where the problem comes. Now let me put it this way. Eschenmoser, really wrote it very nicely. He said there are two aspects to this origins of life question. One is being historical, the other is being ahistorical. And
Mhm.
myself especially, I tend to fall more on the ahistorical side. As I said, my problems are inspired by the origins of life questions, but in the end, they may or may not reveal how historically things happened on early Earth. We have hypotheses. We have, things like RNA world or metabolism first, type of chemistries, but none of them in a historical sense. They all have strong laboratory demonstrations, but you can never extrapolate them back. For example, you can even take the phylogenetic trees that people study and they come to what is known as a LUCA, the lost universal common ancestor. So sometimes I funnily call it the lost, not the last, the lost common. The problem is, everybody wants to have a single point that diverged into this variety. What Peter Gogarten and others pointed out is that there might not be a single LUCA, but there might have been many LUCAs. And therefore, the root is much broader than that. And you can't go from a LUCA to prebiotic chemistry because there is absolutely no connection between the LUCA and prebiotic chemistry.
To rephrase this slightly differently, I think a lot of people might be confused, like, how is there not just one Luca? Because typically, like, in your family, like with humans, you can trace everyone back to the most recent common ancestor of all humans. That's just mathematically, it's impossible not to be able to do that. It's just how things work when you have lineages splitting. But there is a very real possibility that early organisms were much more leaky. They were much more promiscuous, so they could actually transfer parts of their genomes to other organisms a lot. So there was this and bacteria still do this like crazy. Even animals do this a little bit. There's viruses and genomes, and I probably have a virus in my genome that's not in yours, and so on.
Correct.
So there is this leakiness of lineage that exists, and we expect that was much more extreme at the origin or earlier in life than it is today.
Yes, you said it correctly. You beautifully brought up the horizontal gene transfer problem that can confuse things. The other important point is that sometimes many people miss, but it's very real, is that what we are seeing are the survivors. Right? Those didn't survive. We don't have their information and we do not know where they came from. So if you only hang your hat, if you will, on only using the survivors to go back, which is logically what we can do, then you will only get to one point and then you will begin to say, oh my god, that must be the LUCA. And many people have fought battles. I'm not an expert in this area, so I should not really say much, but what from my own experience with organic chemistry and the type of chemistry that I do is there is really only one solution to making a particular molecule. There are different ways to making the same molecule, different ways in a way, and you can't trace that back to the starting material in the same way when somebody else would do the synthesis.
Yeah. So well, this is the other point that you made when you said that LUCA is not the origin of life. LUCA is way after the origin of life. It's the most recent common ancestor of all life forms. So there's a huge gap in not only just our understanding, but our ability to use comparative anatomy to figure stuff out.
Yes.
Comparative anatomy can give you can use bracketing. I can't remember the name of it. Phylogenetic bracketing to try and figure out what the common ancestor trait probably was. But you can't do that. Like, this is I just heard someone say LUCA is the event horizon for life. You can't see beyond it.
Correct.
And it's just like, it's so frustrating. So we have to go to other mechanisms that are far less. I mean, already our picture of LUCA is probably way wrong. And there's just assumptions on assumptions. But then we have to go into this far more complicated thing of like, okay, well, now we can't even use comparative anatomy to figure this out. So we go into the laboratory.
I still remember when Peter Gogarten made this first presentation in GRC, the whole room was in shock. When he threw out the LUCA, the end single LUCA concept out, the whole room was in disbelief, and there was a lot of commotion and questions like, how can this be? What are you saying? What can't we believe? And so these kind of paradigm changing events are the ones that would kind of elevate our understanding and say, oh, we really don't know, and that refrain that we really don't know is something that we need to keep in mind, especially given the historical context and the grayness of that historic. And you brought up a very nice, you said the Luca was an event life in the horizon beyond which we cannot see.
The event horizon, yeah.
Yeah, the event horizon. And that was, driven to me by a struggle that I had for a presentation that I had to make. So I was searching for a picture that would simulate early Earth, for a slide. And so I picked up one picture, and then, it was a little hazy. So I thought maybe can I zoom in to see something? The more I zoomed in, the more pixelated it became.
Right.
And then I realized the problem is not with the zooming in. The problem was that's all the information there is in the picture.
Yes. Yeah. Yes.
Right?
Perfect. Yeah.
So that Luca, that's all the information we have. However, we zoom in, we are going to get noise or signals.
Yeah. Yeah. So that kind of brings us to like what you're doing in your lab is you're trying to figure out it's a lot of documenting how chemistry works, how molecules interact with each other, which you can then put into, as you start to understand it in a very clean setting, you can start to put it into dirtier settings and understand how it works in messier, dirtier environments. But it's like we got to start somewhere. And I think starting clean is where you kind of have to start, right? You don't have to. Other people start dirty, but
yeah. So the question becomes, I have to balance a lot of things that happen when I say I want to have a project. I need to get it funded, and I need people to work on it, right? So the question then becomes, I can get it funded, but then if I get the people to work on it, how are they going to benefit at the end when I publish a paper? Being a synthetic organic chemist, I worry about the type of projects that people will work on and then publish in a good journal and then get a job based on those kind of publications. So I can work on really messy chemistry for all my life and then publish just papers with GCMS or LCMS data. But then I worry about how will that impact the postdocs and the students who work on it to get a good job. So I have to balance both of them. Now, people who work on meteoritic analysis or those kind of things, they don't have to worry about that because that is their messiness. That's an understood thing. And they will get a job based on their analytical skills. But ours is a synthetic lab. That's why we start with certain problems. And there is one caveat that, Eschenmoser taught me, which, is on the top of my mind. It's a top priority. He said, now fashion your projects as you would, and then go ahead and execute them. But then when you look at the result, ask this question: if you remove the prebiotic relevance of the whole project, does it still stand on its own?
right.
If it does not stand on its own, then you are on shaky grounds. Because if you remove the prebiotic relevance and it's still relevant to a field of, say, organic chemistry or medicinal chemistry or something like that, it'll stand on its own and go forward. So a case in point is the very famous Stanley Miller experiment. Very beautiful, and still the most paradigm shifting prebiotic experiment ever, I would say.
Sorry, say it again. Which experiment?
That is the Urey-Miller spark discharge experiment, right? Converting gases to building blocks of chemistry and life. That still stands out. But that same year, Watson and Crick also published their beautiful structure.
Yeah.
Now, fast forward so many years, 50 plus years now. Which paper has had the most impact?
Well, yeah, obviously the genetics. Yeah.
Yes. Why? Because that was so hung up on prebiotic relevance that people begin to question the prebiotic relevance of the Miller experiment. What they forget is, irrespective of the prebiotic relevance, it was a paradigm shifting experiment that showed that gases can be converted to building blocks. And people forget that they want to focus only on the amino acid, and they want to focus only on that, and then they say, oh, it could not have been that reducing, maybe lightning could not have happened in those kind of atmospheres, etc., etc. Those may all be true, but it still doesn't detract from that groundbreaking experiment. But what I'm trying to point out is, if you are so much focused on the prebiotic relevance, it might get washed away, and then people don't give it the credit that is due. And so what we try to fashion out is, I can give you many examples, but what we try to fashion experiments is, by the way, okay, if you remove the prebiotic relevance, does it still make a contribution to chemistry? Does it still make a contribution to organic chemistry? Does it contribute to the understanding of biomolecular structure and function? Those are the kind of questions that we ask. And then the postdoc is safe, because then their paper is recognized by peers and by people who would be able to get them a job.
It's a really interesting thing to think about this. I don't think most people realize this in science, is that the professor has a responsibility to his students or her students to help them not just do the project that the professor wants them to do for the professor's own progress in their career, but also to make sure that it's relevant and attractive to people that are hiring, because there are very few professor jobs. There's tons of chemistry jobs. So you need to keep that in mind. That's really that's clever. That's good. That's a good thing to point out. The way that science is funded and how all this stuff works influences what types of things we can study, which kind of brings me to my question on the status of the field today. I know this is very hard to say, but we don't know what we don't know. We don't know. But how far are we from having a reasonable answer to the origin of life, one that most researchers would find satisfying? They'd be like, okay, the white whale has been killed here.
It's a very good question. And, the problem is that people are, when they come into this field, they already are kind of, going into certain buckets of, how they think about these problems. And very few become open-minded later. So if somebody gets trained in an RNA lab, they become an RNA world person, and it becomes very difficult to talk to them and say, that may not be the case. Or a metabolism first person, they are so hung up on a metabolism first. Or people recently are, latching on to this lipids first model, cells first model, protocells. Very hard to talk to them. And you find these people are, they do not talk to each other in a way that would be conducive for moving the field as a field together. And this has been a problem not only for this field, but for many fields, but it's more acute here, because, as you said, this encompasses a great diversity of techniques, a great diversity of expertise, a great diversity of scientific fields, from astronomy all the way to geology, right? So there is a huge gap in between. So one of the things that we try to do is not be dogmatic. That's the best we can do. We have a fairly decent understanding of what the early Earth provided, how the early Earth might have looked like, what are the conditions, etc., etc. But how that translates into chemistry is still a big question. For example, I will do a very clean reaction based on the idea, okay, amino acids and hydroxy acids existed together. Now, that is a fact that we can take from meteorites, but it also contains a lot of other things. But in my lab, I took only the alpha amino acids and the alpha hydroxy acids, and then we did a wet dry cycle with my colleagues at Georgia Tech, and we showed that the Depsi peptides can go to peptide enriched sequences. So that means you can start from a type of mixture that exists and goes forward. Now, did we do the whole messy thing? We didn't. The problem is the analytical techniques are not there to really go in and tell us how things change. So if you remember, there was a PNAS paper that was published with Facundo Fernandez, where when we even increased a slight complexity of the mixture, the complexity analysis grew exponentially far beyond what we could tackle. So that's the limitation.
So you're saying that one of the reasons we're stuck doing clean chemistry right now is because we can't analyze, our analytical tools just are not there for
And
mess.
even if they are there, what you do is you pick up only the things that you know, you have standards for. And the things that you do not know, those peaks, they remain unknown forever in a way that, so, and that is the problem. So the question is, what are those? So I can identify guanine or adenine in a meteorite. Does that mean that is something that would be useful for life later?
Not necessarily.
Yeah,
Right.
yeah, and you can leave alone the clean prebiotic chemistry that we do in the lab. Let us just take a meteorite that contains most of the chemicals that we think would be needed for life. Nobody can put it in water and shake it and say, voila, life comes out, right? So this is a problem whether you start with clean chemistry or basic chemistry.
Yeah, let me use another analogy to help people understand this too. Is like if you go to the zoo and you watch the animals, how they behave, what they're up to, it might be telling you how they act in the wild, but it might not. And it's very difficult to tease this out. And so we're kind of in the zoo stage of early chemistry where we can only look at these things in captivity in a nice clean little cell, or clean little cage that they're living in. And so we some of the stuff is probably totally legitimate and applicable. Some of it is guaranteed not to be, and we don't know which is which.
You brought up a very nice analogy. I like that because putting the animal in a zoo is what sometimes people do, to understand how they would, they understand the behavior. For example, there are many people who come from metabolism first and minerals first. So many people use minerals. And it's not a bad way to start. But their, criticism, the type of chemistry that we do in clean round bottom flasks is there was no clean flasks 4 billion years ago. So you had to do it on minerals and things like that. And that is a very valid point. I understand. But what they do is they take highly evolved molecules. For example, they will take an RNA that is activated and then they will put it on a mineral and they'll say, oh, it polymerized, therefore RNA world. And my question was, how did that evolved organic molecule get on the mineral in the first place?
Yeah.
So the question becomes, yes, I'm putting it on a mineral that is prebiotically plausible, but the molecule that I put it on is not prebiotically plausible.
The argument they would make is that they're showing a proof of principle that
Exactly. So that means if you're going for the proof of principle, then you can't go for the whole, A to Z, right? You only show the proof of principle that, oh, these reactions can happen on a mineral. That's it.
Yeah.
But you can't say it happened. That's why there is the difference between the ahistorical approach of showing the proof of principle and they claim that it is historical. And the mistake comes when we all try to make the claim that it is historical. And that's when people are up in arms and saying, how can you say that? Or what is the proof?
To think that we are making a model of a possible pathway from normal chemistry to the origin of life. I mean, that if I look at whatever everyone's doing, that's what I would say they're working on building. They're working on building a model for a plausible pathway from chemistry to life. And one of the things that I pointed out in the videos, and this was something that Nick wanted to drive home, is that it's not just that we don't have the exact model, that the history of it, but there are pretty big gaps in that model itself. Once you get to Darwinian evolution, you can kind of say, well, evolution did something there, and there was natural selection, and there was reproduction, and there was variation. But there are parts in the origin of life we don't even have any sort of rough idea what happened. Gaps in the pathway, in the model itself. And I think that's something that a lot of people don't understand.
Yeah. And, just to add to that, the very famous Wohler synthesis of urea, which started with ammonium cyanate. And then he heated that and then made urea. And he shattered the concept of vitalistic force, which said that only living things can make urea, right? So our work is very, in that kind of a flavor, if you will. And the question that we are asking is, how far can science provide answers? Not the full answers, but how far can we try to push our understanding through scientific endeavor and come to a point, yes, we can, or yes, we cannot. So the what we are trying to do here is akin to what Wohler did, but not try to synthesize life in the lab, but can we mix chemicals or bring chemicals in a way that would give rise to an emergent property or a system with emergent properties that exhibit lifelike behavior. Now, that
Right.
itself would be quite a victory in terms of saying, okay, I can mix these things. So, for example, you might have seen our work with the phospholipids, cyclic phospholipids, where we
Yes.
create cells and protocells which begin to divide and which begin to, peel off and give smaller structures. They almost act and look as if they are alive, because of the room temperature condition itself that provides enough energy for them to fluctuate between different structures and things like that.
Mm.
So now that is not life, definitely not life. But the thing is, it begins to show us what sort of dynamic behavior that can
Okay.
happen with the given energy that is provided at room temperature for a certain system. Now, if we
Mm.
can use that and build on that to do certain other things, couple it with metabolism or couple it with RNA replication, which is
Mhm.
what Sheriff Mansi did, with RNA replication. Those are the kind of beginnings that we are looking for. And these are in no way saying that, oh, we have created life, but we are beginning to understand what are the rules. We begin to see that these kind of properties emerge naturally.
For people who don't know the Voller experiment, it used to be thought that all organic molecules could only be created by life through some sort of vital force. And then he made urea, which is an organic molecule, just by mixing the right things together with the right amount of energy. And he's like, whoa, wait a minute. This is actually just, this is
Yep.
chemistry. Um, and so that there, but that's just urea. So you could say, well, he proved there's no vital force producing urea, but what about all the other cool things that life does?
Yeah.
And you guys are you're checking a bunch of stuff off the list, like, oh, we can do this without, we can do this without, we can, these molecules are very capable. The physics here, the chemistry here is capable of doing things we did not imagine were capable before. What's the last discovery and origin of life that you were not involved in that you're like, wow, that's impressive?
Most of my amazement comes more from discoveries from, say, early earth conditions that people study, or from exoplanets that they study, because those really change or challenge what we understood to be possible. For
Yeah.
example, there was a recent paper got a lot of press attention where they said that they found certain types of organic molecules which were in the planetary disk. So
Mm-hmm.
usually people usually say methanol or some nitriles and simple nitriles and things like that. But this went way beyond the stage of that means these give the actual possibility that the organic molecules could accrete with the planet formation at the time of planet formation. And
Oh.
for me, that changed my level of appreciation from, oh, I see molecules in the interstellar space, etc., etc., to actually molecules that could be present at the time of planet formation.
Oh, that's cool. I missed that paper.
I will
Uh,
send it to you afterwards. It's
yeah,
a
yeah.
very nice paper, and
Yeah.
and the paper is written also in a very nice manner. They don't make any claims to origins of life or anything. They have really done a nice job of saying what they observe and what the implications might be. But the thing is, the fact that they could observe these molecules closer to a planet formation stage gives us hope that there might be more molecules that could have survived and become part of the type of chemistry that we think would be possible. Right
Yeah.
now we are debating, for example, there is the cyanide based chemistry versus the carbon dioxide based chemistry. And carbon dioxide is known to have existed and, very well documented on early Earth, but cyanide not. So cyanide has been more of a model. People have modeled, oh, if there was a big hit on Earth, there would have been a certain reducing atmosphere for a certain period of time. It would have produced cyanides, etc., etc. So there is no real evidence of cyanide on early Earth, but so people then say, oh, we'll use only CO2 because we know CO2 was there. And people say, no, but cyanide chemistry is so rich that it gives you all these things that you would want to start the type of chemistry needed for life. So that is the dichotomy that you see, in this field, to work with what we have. But then that really requires really harsh or unrealistic conditions that really doesn't get you much. Or you work with molecules like cyanide, which are energy rich, which gets you a lot of things, but then their origins and their presence and their sustenance on early Earth is doubtful.
I want to do a quick aside here.
Yeah.
I don't know why this popped in my head. There's a lot of criticism of the field that's valid, totally valid. And then a lot of it that you can tell is supercharged by something. And in America, we know what that something is. It's people's religious views. A lot of people feel like if we have a natural explanation for this, then that erases the possibility of God being involved. And I know that at the Center for Chemical Evolution, when I was working there was a bunch of Christians that were there, the Origin of Life chemists, and they didn't have qualms with it. I'm curious. I don't know if you want to talk about your religious views or how you if this has ever been like an issue for you personally, but only if you're comfortable talking about
No,
this.
no. I've been asked this question many times, and for me there has been no personal qualms, because the type of philosophy that we were brought up in actually allows to a level where there is no creator God. Probably this Hindu philosophy is the only religion, and of course Buddhism is an offshoot of that. They go to a level where there is no creator. You know, it's uncomfortable for many of the levels. There are many levels there. So there is a level where there is a creator God who will give you good things if you do good things and punish you if you do bad things. Then there is another level where that creator God is not separate from you, but who resides in you, who is in you. And then there is another level where there is no difference at all, and it is I won't say it is one, but it is non-dual. There is no duality there. So in that sort of a level, they don't talk about creation, they talk about only manifestation. So there is only manifestation, but there is no creation. So for example, the type of example they would give is mud. Pot is a manifestation of mud, right? So but for a pot you needed somebody to create the pot. So that means there is a creator who created the pot out of mud. But for us, we would say, no, pot is just a manifestation in space. Mud is in a different shape in space. And it can be formed, by nature. So you might have seen these kind of formations that people have put. There are these stone arrangements that naturally happen when water washes back and forth, and
Oh,
it
yeah.
sorts them on their sizes, right?
Yeah.
And then it looks as if, oh, somebody must have done it,
Right.
right? So I can give you a very a beautiful example that drove the point to my own children. So I had taken them to the San Diego Safari Park. There was a San Diego Safari Park. I do not know whether it still exists, where you can sit on a monorail and it'll take you around, and you will see different animals in their so-called natural settings. So when we are going past a mountain, we stopped for a while, and the driver said, just keep looking at the mountain. And suddenly figures started moving in the mountain. And these were mountain goats which really beautifully blended with the scenes that you could not see them until they moved. And so the question was, my children, how did this happen? How can the goats know to have the same color as the mountain?
Yeah.
Right? So luckily for me, I had watched Carl Sagan many years ago. And so I tried to explain to them that it is a form of natural selection. That means there are different colors, and the colors that got picked up by the praise were the ones the praise could see, and then the praise that could not see the colors, those survived.
Right.
They accepted it because their father told them at that time, but it was not very convincing for them. So when I came home, I played a game with them where I put a plastic sheet of a color, I forget which color. Then I took marbles of different colors and put them there. And I said, you both of you run from one side to the other and pick up as many marbles you can.
Yeah.
The one who collects most marbles wins. So they ran from this side, they collected the marbles, and then they ran back from this side, collected the marbles, and then they said, oh, we are done. Are you sure? I said, are you sure? They said, yes, we are done. Then I lifted the thing, and the marbles, which had the same color as the sheet, fell black, fell out.
Yes.
And so that taught them that was a natural selection. Now, you can't say the marbles knew how to blend it, right?
Right. Right. Yes, that's beautiful. That's excellent. One more question on the status of the field today.
Yeah.
Are there any specific problems that everyone seems to be racing to solve? So, areas where there's pressure to publish fast so you're not scooped by a different team? Or are different groups focused on totally different problems? And I guess another thing that on the side in there is you talked earlier about there's kind of like some closed-mindedness in certain parts of the
Yeah.
field or like aggression. Do you think that's ever valuable? I mean, that like the aggressiveness, the competition, is that ever valuable? Is another thing to throw in there.
It might be valuable to motivate yourself, but it becomes really, I would say, a negative point when you publish something and then in that publication you deride the other hypothesis in such a way that it makes the people not appreciate your work, you know.
Right.
So that's what I see happening in some of the papers, especially written by some leading figures or leading personalities who have very strong opinions. They will publish a paper in a way that they say, here it is what we found, and then they will go into a paragraph of discussion where they will just dismiss the other concepts. There are people who have, you know uh, uh, compositions, composomes, and then they will deride the RNA world. And then there are people in the RNA world who publishes an RNA world, and they will deride the metabolism and the,
Right.
um, you know, those kind of things happen. I would rather focus on what the paper does. So one of the things, again, Eschenmoser taught me was, don't stop reading a paper because you don't like the personality or you don't like the hypothesis, because you may still learn something from their paper, interesting chemistry or interesting ideas which might stimulate you. So, but I would say largely the field is trying to, because each field has run its own way, and probably they are butting their heads against certain kind of wall, if you will, and then they come back a little bit. They give a little leeway to, yeah, coevolution of proteins and RNA, and then the coevolution of this and that, and then, but they still try to go back to their, you know, original roots.
Much of the research that scientists do is debunking their own hypotheses or debunking the hypotheses of other people, and sometimes those can be very beloved hypotheses. So, aside from the foremost reaction, which we're going to talk about here shortly, what is the biggest no that you've shouted at the others in the field during your career, and how was it or how is it being received by the field?
So that was a very difficult time for me, because I was working with Eschenmoser and we had worked on the alternatives to RNA, trying to understand why RNA, not how. That's a very different question. Why RNA? Why is RNA so special? And so during that time, we studied various alternatives to the nucleobases. And one of the molecules we studied was orotic acid. And orotic acid has a very special place, because if you go and look in biology and life in all the animal, life throughout, orotic acid is the only nucleobase that nature makes as a full molecule and then attaches it to ribose. Okay?
Okay.
Adenine is not made fully, guanine is not made fully, cytosine is not made fully, uracil or thymine, nothing is made fully. They are all built atom by atom on ribose. Okay.
Oh, okay. I
Yeah.
didn't know that.
Oh, yeah, it's a big difference. You go and look into the biosynthesis, adenine is never made fully as a molecule, then attached. There is a salvage pathway. Once it is made, it's so difficult to make that nature tries to preserve it and brings it back like a salvage pathway. But
Oh, okay.
the only molecule that nature makes is orotic acid as a nucleobase, and then attaches it to ribose and then decarboxylate to make uridine. That's the first step in making uridine. So uracil by itself is not made by nature. Okay. Now the question is for me was, if orotic acid is made by nature, why is it not in RNA? Why does it have to decarboxylate and make uracil and only use that? So when we looked at the literature, nobody had made orotic acid containing RNA to study their base pairing properties. When we did that, what was shocking to me was that I had an inkling it would not pair, but it was the weakness was so great that even a single insertion would destabilize the duplex and destroy the duplex formation. of the two strands.
And how long of a duplex?
These were all 12 mers and 10 mers, which usually would not, usually, even we made a 12 mer, I think, 12 mer and a 16 mer, and it was surprisingly weak. And, so the question, I took it to one of my colleagues who was at downstairs, a famous RNA world person, and I showed him, hey, look, this orotic acid is not functioning. And his immediate response was, oh, it's not surprising. Nature must have chosen a better base pairing base. And that immediately out of my mouth came, but when did you know it was that a better base pairing system? You had to make an oligomer that long, and you had to wait for a complementary base pairing partner. So when it is just the nuclear base, you could not have made the selection.
Right.
Right. So why are we getting excited about a nuclear base in a meteorite when we do not know whether it will be selected or not?
Yeah. Yes.
Right. So that set me on a very difficult pathway of dialing back the involvement in an RNA world type of concept, because the RNA world can only emerge at the level of an oligomer where there is base pairing and there is information, but not at the level of a single nucleobase or a nucleotide or a nucleoside. And that set me thinking about what would be the alternatives. And so I became not, I would push back against the strong claims of an RNA world. It's not that RNA world is bad or anything. It's just that people who say, they'll make these long sequences, they'll make an in vitro evolution, and then they will say RNA world without even giving a thought to how that, single block was synthesized. They will use nucleotide triphosphates in excess, and then they will say, oh, RNA evolved. But then where does your triphosphate come from? They'll say, oh, it's not our problem, it's a prebiotic chemistry problem.
Yeah.
So those kind of things there. And that is when I started, and there I received a lot of pushback.
I want to point out to people, I did a video on the RNA world. In the end of it, I show several times I paused to explain that there's unsolved problems. While the basic idea of the RNA world does seem to give us a promising pathway to the origin of life, it's still very much a work in progress. As mentioned, one of several unsolved problems is, how did nature get backbone binding to function without the special enzymes or lab techniques we use today? And that's Ram coming through in my videos. So people know. He's like, the RNA world's important, but if you're a purist about it, you're going to miss these problems and so on. So yeah, that was very helpful. And that was your coaching that really helped me understand this. I mean, Nick pushed me to talk to you about this because he's like, Ram knows why. He'll explain it to you. We have this problem in science communication. Oh, scientists just solved a major mystery in the origin of life, and you look at it and it's
not.
Well, it's not really, but yeah, this is incremental progress. This is interesting information, but it's not.
In the same vein, I would also say I've gotten pushbacks when I also argued against a pre-RNA world. The problem with the pre-RNA world is the following, and I put it in paper as well. The problem is this. So the pre-RNA world, people say, oh, making RNA is difficult, which is true. The nucleosylation problem is still there. And then they come up with alternatives and say, well, okay, we can make these alternative nucleosides much better. Then that's fine. But then how does that pre-RNA make RNA?
Right.
That they have not solved. So when I talk to them, they will say, oh, that comes down later, but that is the same thing RNA world people do. How does it solve? Oh, it comes down later, evolution takes over. So the problem with the pre-RNA world is also the same that RNA world faces, because the pre-RNA world will say, oh, we can talk to RNA, we can transfer information to RNA, we can replicate RNA, that's all fine. But how do you still make the nucleosides of RNA, which was the initial problem in the very first place?
Yeah. Well, but yeah, if you were to talk to Nick about this, he'll say, like, yeah, we know that that's a problem, but we're working on this part right now.
Yeah.
Because you can only do things one step at a time.
Correct. But what we can't do is the pre-RNA world people should not fall into the same trap that RNA people fall into. So that's my caution there.
Right. Yeah. I mean, yeah, probably all this business about worlds is kind of it makes unnecessary mutual exclusions, right?
Correct.
Or things were obviously messy. And, you've done some work showing how selection happens in a messy environment. But when I say messy, it's a lot cleaner than it would be.
Oh,
but,
it's a clean mess. It's a clean mess.
yeah. So that's Ram the naysayer. But let's continue on Ram the naysayer.
Yeah.
With the Formos reaction, which is a paper a couple months ago that sparked this conversation here. I wrote down something that might be useful for people that are new to this. So the Formos reaction has long been a favorite reaction proposed for the origin of early sugars, such as ribose in RNA. Ribose is it's the sugar that's in RNA. And in your recent paper, let's see if I can pronounce molecule names. Abiotic aldol reactions of formaldehyde with ketosis and aldosis. Implications for the prebiotic synthesis of sugars by the Formos reaction. And you essentially say that we've been barking up the wrong tree. The Formos reaction is not what we hoped it was for origin of life.
Yep. That's correct. So go ahead. You said it best.
Yeah, I guess briefly, simple as possible.
Yeah.
What is the history of the Formos reaction in origin of life research, and then why was it a favorite, and what problems did we hope that it solved?
So the Butlerov, when he discovered the Formos reaction, had no prebiotic chemistry in mind. He was just trying to make sugars, and he actually tasted it, and it smelled sweet, and that's how he knew he made sugars. Now it was co-opted much later by the origins of life community, because formaldehyde is such a simple molecule. It's like so if you take ribose, it's 5 formaldehydes together. If you take glucose, it's 6 formaldehydes together. So that became a model saying that if you can start from such a simple molecule and make sugars, maybe this was the way that it started on early Earth. And so then people started worrying about whether formaldehyde would be available on early Earth. Again, this is one of those molecules like hydrogen cyanide. There is no proof that it existed on early Earth, but since it gives all these kind of things, you come up with models where it could be generated, it could possibly exist, and things like that. But these kind of molecules have been seen in interstellar space. You can see a lot of these spectrums of that. So people assume that it should have been available on early Earth. But the problem is not, for me, was not with the availability or its reaction, but whether there was really ribose in that mixture to an extent that would be useful for any chemistry. So when we started, we didn't start actually with the Formos reaction. We started with our glyoxylate chemistry, which was much cleaner, and we are writing a paper on that. But then we wondered, how messy is this Formos under these same identical conditions? And so that is when we started studying the Formos reactions under milder conditions. And when we looked into the literature, there was very little systematic study using NMR as a tool. People have certainly done NMRs on Formos reaction, but that was under different conditions or different later stages, only the end stages and things like. But we really wanted a time dependent analysis of this, and so we went into this very benign conditions where we thought, because the actual Formos reaction is a very harsh conditions, high pH, calcium salt, high concentration or low concentrations even, but very high pH and heat, and the whole thing goes to pot. So if you let it go more than 3 or 4 minutes, it becomes dark brown, dark black with time, and then becomes very difficult to analyze. And then there was the claim by Steve Benner.
Are things sticking together and branching? Are they kind of melting together?
It's like almost like a polymer that is being formed. Okay. And so but what they do is, many people did the analysis within the first few minutes, and then they say, okay, we can see these kind of sugars mostly by LCMS after derivatizing the mixture. And what we faced a problem was, if you do the derivatizations, these derivatization conditions are not innocent. They can also change the product ratios, because they are usually under certain highly acidic conditions or heating conditions, which can change the product composition. So we decided to take a non-invasive approach by using NMR and just following the species as they form and they react by using C13 labeled formaldehyde and C13 labeled corresponding reacting components like glycolaldehyde or glyceraldehyde or dihydroxyacetone. And that is when we began to see the rise and fall of species, as you would expect in an aldol reaction pathway. And to our surprise, we never saw any carbonyl migration. So for the formation of ribose and the formation of other sugars, you need to have a carbonyl migration that would allow you to go from a ketose to an aldose. That is, the ketose is a carbon which is having a carbonyl group which does not have a hydrogen attached to it, whereas an aldose has a hydrogen attached to that carbonyl group, which is like ribose and things like that. And so that was a big surprise for us, and so we never saw any retroaldol reactions. We never saw carbonyl migrations, which are at the essence of the so-called Formose reaction for this to happen. Now, given, our conditions were very benign, very normal room temperature. But if you heat it, which is what the Formose reaction does, you begin to see this, but then the reaction becomes quite complicated and quite messy, that there is really no ribose in there,
Yeah.
And the problem was that, Steve Bennett published a paper claiming borate would give you ribose. But when we repeated that reaction, and when we actually went and looked at Benner's paper in great detail, there is no yield of ribose. There are some peaks, very, messy peaks that, one is pointed as ribose, but we couldn't. But there is plenty of information on the branched sugars that are formed, the yields on the branched sugars. So what the Formose was doing was leading to more branched sugars than any linear sugars. It does not stop at the linear sugars, which is what you would want for a ribose to become part of RNA.
Wait. So I'm not familiar with branched versus linear. When you say linear, do you mean like a single monomer of sugar that's open? It's not looped?
No, that is a cyclic. That is acyclic versus cyclic. That's a different story. What it is if you take and you draw a line, a zigzag line, right? That's a linear zigzag line. But if in that linear zigzag line you begin to branch or draw, hanging things out of it, that's a branched. Okay. So what this Formose does is it does not produce the linear zigzag, but it rather produces a branched, zigzag
Of monomers.
of monomers, which are these are what they call, ketoses. Branched ketoses are formed, not branched aldoses. And
So the number of atoms is correct for a ribose, but they're not arranged properly.
they are not arranged properly, and it doesn't stop with a C5 carbon. Ribose is a 5 carbon sugar. What this ends up in a 7 carbon sugar. It ends up always in a 7 carbon sugar, which is completely branched. And so we did our best, not to be biased. We didn't want, we really didn't anticipate this going in. So we tried to find out whether there would be other ways to mitigate or, make more linear sugars that would come from the Formose type reaction. But there was, in our hands and under our conditions, our analysis showed that it is not possible. And so that's the biggest point, is that the so-called Formose, even under very mild conditions, does not give you the ribose that you're looking for.
Right. So in your mind, this scratches the Formose reaction or very similar reactions off the list for the origin of ribose.
And under the type of conditions that we have seen, this does not mean that there might exist a special condition under which this can be controlled. So one of the caution that we give in the paper is that this should not shut off the avenues for such research, but it should shut off the claims that the Formose reaction has been a source of RNA, which is what currently exists in the literature.
Okay, so the claim is premature.
Yes.
But yeah, I mean, I guess that pulls up an interesting thing. We can conduct a test. We can just miss the correct thing when we do a test. So we can think that we've debunked a hypothesis when in reality we did not. There's tons of ways to do that. There's tons of ways for us to confuse ourselves when we're doing research, especially when it's chemistry and when there's messes involved. So this is an interesting problem. We would like to be able to just scratch things off and say, oh, we've been barking up the wrong tree. Let's go to the next tree. But you kind of need someone to linger there just in case. Editor's note, at this point in the conversation, Ram and I started talking about people we know in the field and how they specifically reacted to this paper in private conversations. And we quickly realized, we're talking about private conversations here. We should not put this part in the public video. But I can summarize things and just say this. Even some of the people that Ram thought might be frustrated by his new paper were not. They seemed very grateful for it. Everybody seems to be incorporating this into their thinking now. There are people holding on to a couple of things that Ram maybe didn't fully check off the list for the foremost reaction, but overall, a very positive response from people in the field. But we will let them respond publicly in follow-up papers or if they do their own videos on this topic and so on. So back to the conversation.
It has been largely positive. I have not received any negative feedback. In fact, I have received requests from many teachers who say, can you send me a copy of this paper? We would like to incorporate this in our lessons, in our teaching. Sure. Because, usually people say, oh, foremost has been this, make sugars, and then they go to the next and next and next, right? But now they have a paper which says, no, that's not correct, so they would like to present that viewpoint as well.
So it does look like it looks like people took this seriously, because one thing that does happen in science too is there's so much stuff being published, people don't read it all. So you can get ignored for just the sheer reason that there's volume, there's too much for people to do. I guess that kind of closes it off for Ram the naysayer. Sometimes you tell people no. That's part of the job of any good scientist, and you seem to focus on that a little more than others. But it's There's also Ram the Pathfinder. There's this new paper that you just published on selection. Let's talk about that. Can you describe that to a lay audience?
Yeah. So the question now becomes, let us say we do have a reaction that gives us a mixture of sugars.
Mm-hmm. So you got a mess of different sugars.
You have a mess of different sugars. And the question is, are there ways to select a particular sugar without, I'm not being teleological here. It's not that I want a particular sugar. Is there a mechanism to select the type of sugar from this reaction mixture? And so
is there something in nature that will just sort them just the way that rock sizes get sorted in a wave?
Correct. Yes, exactly. Very nice way of putting it. Yes. So the question was, is there a chemical that would do that? And when we started looking at this reaction of a phosphorylation reaction where you attach a phosphate to a particular molecule, what we found was this molecule that we use, which is called the diamidophosphate, which is nothing but a phosphate molecule where we have replaced two oxygens with two amino groups, allowed us to do that. And, it turned out that ribose was the one that got selected mostly from this reaction. And in a fashion that allows this molecule to react further, probably producing nucleosides, which is the next step towards the nucleic acids. So here we tested the 4 pentoses. We didn't test all the sugars because we had some idea from previous papers that, for example, 6 carbon sugars would not react. So that I know from my own experience in my lab is that when we try to react 6 carbon sugars, because the 6 carbon sugars, they close and form the cyclic structure, which is a hexopyranose structure, they don't open up very easily. And for this phosphorylation to happen, you need to have an open form. And it so happens that out of the 4 pentoses, ribose is the one that has the most open form.
Yeah.
So this molecule took advantage of that fact, and then ribose reacted much faster than the other 3 pentoses, and it selected itself out by a factor of almost 6-1. So that means for every 6 molecules of ribose that would react, only 1 molecule of the other sugars reacted. So that means there is a overwhelming preference for ribose to go forward and leaving the remaining sugars behind.
Let me try to explain this again too. And I might have understood this wrong. So what I'm hearing here is that, so you've got these sugars that are just drifting. So you've got monomers, single sugars drifting. There's a bunch of different types. Then you have phosphorus, and that can attach to these sugars And when you have DNA or RNA, you have the sugars, and they're attached with phosphorus to each other to make a chain.
Correct.
And what you found is that the phosphorus likes to attach to ribose more, likes in quotes here. It attaches easier to ribose than it attaches to the other ones. So just through this normal process of phosphorus being added in the right conditions, it's going to start making chains of ribose, and it's not going to make chains that involve the other.
Yeah, that's right. It gives rise to the potential of making chains from ribose and not from the other sugars. So if you will, so this phosphorylation is a natural selection for ribose.
Yes. Yeah. Right. Natural selection and just this it not in the Darwin sense so much, but more in the rock sorting sense.
Correct. More in a chemical reactivity sense. It naturally reacts with ribose more than the other sugars.
Yeah. That is cool. That is cool. And for those who don't know, Ram's work is filled with these. He keeps finding these ways that nature can sort, and it's really cool to just go through his work and see all the discovery on discovery of him finding this. It's kind of, I would say it's one of the themes in your work.
Yep.
Yeah, that's really cool. That's already published, right?
Yes, that's
Or
already
did you
published.
send me?
Yeah.
Okay, yeah. Does this have any implications for the foremost reaction that you previously said is not so good? I mean, does this maybe pull it back into being a possibility again?
So no, what does this is it increases the pressure in order to find different source of sugar molecules.
Yeah.
It still might be the foremost reaction under conditions that we have not discovered before, which would do this. I'm open to that. It's not that I'm shutting it off. It's just that I do not know at this time what would be a good way to convert formaldehyde into ribose in a fashion that would be uh, um, productive, not the typical classical foremost reaction where, or in the presence of borate, where you really don't get any ribose in a meaningful fashion. I still do not know that. One other possibility that a work that was done with Nick had was that you can build higher sugars, but then you can break them back to ribose, which is what actually, if you look into biology does not make ribose directly. There is no C3 plus C2 reaction or a C1 plus C1 plus C1 plus C1 plus C1 that gives ribose. What actually nature does is it takes a C3 sugar and another C3 sugar makes a C6, and then the C6 reacts with another C3 sugar to make a C9. These are all known as transketylation reaction by enzymes, mediated by enzymes. And then the C9 breaks down into C5 and C4. That C5 becomes part of the ribose. This C4 joins with another C6, gives you C10, which breaks down into two C5s. Right. So that's how nature makes the C5s.
Yeah, that's how cells make it, right?
Yeah, that's right.
And as you pointed out earlier, like we can't. I mean, maybe that means that nature was doing that in some environment somewhere too, in a similar pathway, or maybe we are looking at lions in a cage and they behave totally different out in the wild. So the cradle of life, wherever that was also making ribose in some completely different way, and life then ended up also finding a way to make this too. This is such a complicated puzzle.
You begin to see that. The question is, how much of biology can you take and how far can you take it back before it breaks down? One of the examples that I give is, for example, I use myself saying, oh, I come from a country, where, there are people in villages and they live in, mud houses with thatched roofs. And then you come to New York and you suddenly see this beautiful skyscraper and you wonder, how did they make it? And you are coming already biased by how the mud thatches house was built. So you must think, oh, they must have put the first floor, then the second floor, then the third floor. Oh my God, it must have taken them a long time. And then somebody, comes along and says, no, there were cranes. The cranes were, just pulling things and pushing things off. But then this person says, but I don't see the evidence of cranes. There are no cranes here. It's only the building. So what we are looking at is at a finished product now, many millions of billions of years of evolution, and many of the things might have vanished and we do not know how to look for them. How do you look for the evidence of cranes in a finished building?
Right.
You can't.
Right.
So
That's brilliant. Yeah.
yeah, so the same way, and people say, oh, that's a very good example, but that's not really. But then I say, look at the pyramids. We are still wondering how they were built.
Right. Right. Well, there was another there's another thing that I think about sometimes is imagine like an alien finds a spaceship with humans in it.
Yeah.
And they look at that spaceship and they want to, and maybe the humans are all dead or something. So like, oh, I wonder where these things came from. What was their cradle of life? What was their homeland like? You have some clues in there, like there's oxygen and there's an atmosphere inside the ship, but these are highly trained astronauts. They're wearing really weird clothes. Their shelter's totally different than the shelters that humans live on Earth. The ideas you would get just from having them,
Yeah.
when you would draw these pictures of the ancient or the distant planet where they come from, and it would be nothing like
Yes.
what it actually is. So, and that's what a cell, I like to think of a cell as a spaceship. It left the cradle of life a long time ago.
Very nice. Very nice. Yeah.
And it carries with it probably parts of that cradle on the inside. I mean, we can really see this with us. We have like seawater inside our bodies basically because we came from the animals that lived in the ocean, and we still carry that with us. It's still in our eyes, and we're still wet on the inside. So you can see that we're kind of like fish in a spacesuit living on land. But so there are clues, but you just don't know which of those clues are real clues and what's
Correct.
misleading. It's
Yeah.
very difficult.
Yep.
Well, I guess in my final question, so the origin of life must have been a big, messy series of events. How do you take a specific finding like this one you just made from a relatively clean lab setting and place that in the chaotic context of the real origin of life? Is this even possible, or are we fooling ourselves to think we could ever make sense of such a wild process?
Very good question. Again, very difficult to answer. But the best answer was, for me, was not a definite answer, but what Eschenmoser said. He said, as a synthetic organic chemist working with such clean chemistries in the lab, the best you can hope is not to find out how it happened or did it happen, but to show it can happen.
There we go.
So that's where we are.
Perfect ending. Perfect conclusion. Yeah. This was awesome.